Hi, I'm Mikayla Truong, and today I'll be walking through the manual backbone assignment using POKY. First, I'm going to generate a practice set of data by typing in the two-letter code ME. I'm going to be using the protein ubiquitin, so I'm going to use this BMRB entry number, 6457. Next, I'm going to use the solution experiment state. And since we're just doing the backbone assignment, I'm only downloading the backbone data. Then I'm going to apply a level 2 noise level. However, you can use any level you'd like to practice with. Next, it will ask you where you would like to save the files. I have made a file called backbone assignment where I'm going to be saving it for today's video. However, you can download it anywhere you would like. From here, it will take a few minutes for all of the files to download. Once all of the files have downloaded, it will automatically open in POKY in all these windows on top of each other. However, for backbone assignment, we'll only be using the NHSQC, HNCACB, and the CBCA-CONH. So we are going to close these extra windows. Now I'm going to spend some time organizing our workspace. I'm going to pick one of our windows and I'm going to set it to the dimensions that I would like. Ensuring that it is selected, which you can see here, I'm going to type in the two-letter code US. This changes each of the windows to have the same dimensions, making a more uniform workspace. Now I'm going to type in the two-letter code VT. This opens the view settings. From here, I just want to turn on the axis atom names for each of the spectra. So I'm going to make sure that's checked and then press apply. You can easily change which spectra window you're looking at by pressing the view up here. From here, I'm going to just turn on all of them. Once you apply it to each of the windows, you can see that the axes are now labeled, with the 3D data having hydrogen, nitrogen, and carbon, and our 2D data having the nitrogen and hydrogen. Now I'm going to zoom into our data for a closer look. You can do this by holding the Shift button and pressing the plus button on your keyboard. To zoom out, you just simply press the minus button. All of our peaks are very small and kind of hard to look at, so we are going to change the contour level a little bit. You can do this by pressing the quotation and apostrophe mark button. This makes the contour level go up, making for more noise and larger peaks. If it is too much and you would want to go down, you can press the colon or semicolon button. And we'll be adjusting this for all of our spectra just for easier viewing. Once this is set, we are going to try to pick each of these peaks. But instead of doing it manually, we are going to use the automated peak picking system, which also known as APES. You can open this by typing in the two-letter code AE. It will automatically open the file window. What it is asking for is a file with your sequence information. Since we are using a practice set of data, this was automatically downloaded in the location that we had previously picked, here in the backbone assignment folder. This data can be found in the bmrb number.fasta file. However, since it is this extension, we have to go down here and change it to the second option for this file to be available. From here, we can select it. It will then automatically open the APES peak picking window. Here you can see that it automatically put in the number of residues as 76, which is expected for ubiquitin. Next, we are going to simply put in all the file types we have in the correct slot. For example, we are going to put in our NHSQC file into the NHSQC slot. And we'll be doing this with all of our data because we want the peaks in all of our data to be picked. From here, we're going to just run APES. It will automatically ask you if you want to do the iPine automated assignment. However, since I'm showing you how to do manual assignment today, I'll be skipping this step. Now we simply wait for the program to finish running. Once the program has finished running, this window will open, stating that it has finished. We're just going to simply press OK and close the apes window. Once the program has finished running, we can see that a lot of the peaks have been picked, although it has missed a few, which is expected for an automated system. However, it did do a lot of the work for us. Now we can start our backbone assignment by selecting one of the peaks in NHSQC. But we have to ensure that the pointer mode is in this first box, which is the selector mode. I will be picking a random peak to start our backbone assignment. Once it is selected, you can see a small box around the X. From here, I am going to type in the two-letter code VC, which will center the peak in our window. Making sure that it's still selected, I'm going to press each of the other windows and type in the same code VC so that each one is centered at the same point. We can now see that each one is centered on this peak across all of the spectra. From here, our next step is to assign this peak. You can do this by typing in the two-letter code AT, which will open this window. Here, we are going to name this peak X100. And the reason we are doing this is because X does not represent any amino acid, so we will not be confused with this assignment. And we know that the ubiquitin sequence only has 76 residues. Since 100 is outside of that range, we can see this as a placeholder name. Here I am just typing in the atom that is horizontal, and by applying, we now have named this peak. However, this ornament is very small. You can increase the size of the ornament by pressing Ctrl M. And this will make it larger for easier viewing. Now we are going to open our strip plot by typing in the two-letter code SP. Once this is open, you will go to Show on the upper bar and select Strip Spectra. For our strips, we are going to use both CBCA-CONH and the HNCACB. So we are checking both of these boxes. I will also be turning on the spectrum name under each strip so it's easier to follow. Now, we will make sure that our peak is still selected before adding the peak by pressing the plus peak button in the strip plot. We can see that this peak has now been added under the CBCA-CONH and the HNCACB spectra. Before I go into the next steps, I want to explain what we will be looking for. Since we are using these two spectra, we need to understand what each one will be showing. For the CBCA-CONH, it is known that there will be two peaks for each strip, or this is at least expected for our strips. And what these two peaks will show is our I-1, with I being the residue we are looking at. So in our case, the residue we are looking at, we named X100. So in the CBCA-CONH strip, the two peaks that show up should represent residue number 99, which is our I-1. Meanwhile, the HNCACB will show the carbon alpha and the carbon beta for both the I-1 and the I. So the I-1, which is again our 99, should align with our peaks in the CBCA-CONH because they should be representing 99, the same peaks. Then the additional two peaks that we expect to see on the HNCACB strip should represent our I, which in this case would be 100. So when comparing these two strips, we can identify which peak belongs to which residue by seeing whether they align and what is left over. So now we're going to look at our actual strips. The two plus signs on this button represent the zoom in, which we'll use to zoom in on our data more closely. What we can see here is that when looking at our CB, CA, CONH, there are two peaks, just as we had expected. And you can tell that those are the two that we're looking at because when you put the cursor over the peak that you have selected, the line runs directly through those two peaks. Additionally, the HNCACB, we're going to zoom out a little bit for this one, has the expected 4 peaks that we were expecting. 1, 2, 3, and 4. And additionally, we can see that these two and these two align, while this one aligns with nothing and this one aligns with nothing. Knowing the information about each spectra that we had just gone over, that would indicate that this and this peak is part of the I-1, so our 99, along with this one and this one, while these two most likely represent our 100. Another piece of important information when looking at these strips is we can see that there are two different colored peaks in each of these, with our green ones representing a negative signal, which more often aligns with the carbon beta, while the red one represents a positive signal from our carbon alpha. So when looking at these two aligned, this one being green indicates that this is the carbon beta of our I-1, so our 99, while this is the carbon alpha of our 99. In order to not confuse these, we are going to spend some time labeling each of these. As you can see, these ones are missing peaks from our apes. In order to add a peak, simply hold Control and select. And to make sure that that peak is centered on the center of the peak, you will type in the 2-letter code PC. And we'll be doing this on this one as well because it was not selected. Now we will begin typing in our assignments for each of these. So again, to open that window, type in the 2-letter code AT. Okay, let me try that again. AT. There we go. So this one we have identified as the X99 carbon beta because it aligns with the green. However, since we are looking at the X100 on the NHSQC, that is the amide group that corresponds with our assignment. So here we would actually write X100 along with the hydrogen X100. And you put in the corresponding atom, but we know that the carbon is actually the X99 carbon beta, which we will signify by CB. And we are going to apply this. Now you can see that it has been labeled. And then we'll do the same here. This is our X99 carbon alpha, which is seen by the red peak. So I now select that peak and open the window AT. From here, very similarly, we're going to type in the information we have about that peak. This time, we'll be typing Ca for carbon alpha. And we'll be doing the same for these. Now that we have labeled the peaks for our I-1, we can deduct that the last two that are in line with our peak from the HNCACB would represent our I, so the X100. Now we will label these the same way that we've been labeling all the other peaks. Here, we are still in the same amide group, so it'd still be the X100 for the nitrogen, but now the carbon would also be the X100. Since the peak that we are currently selecting is red, we can say that it is our carbon alpha. This final peak was not picked as well, so I'm going to zoom in and select it, ensuring that the peak is centered by typing in the two-letter code PC. Now we can assign it. Now that we have put in all the information that we know, we are going to move forward along our sequence. So we will be looking for our X plus 1 or X 101. And this can be done by selecting our two X100 peaks, which were the two that were not aligned with anything in the HNCACB. You can do this by first selecting one as you usually would by clicking on it, then holding your Shift key on your computer, selecting the other one as well. Now, we are going to select the CB-CA-CONH spectra, but not on any peak, just on some empty space, which you will have known that you have completed by looking at the fact that a green outline has shown up. With this, we are going to press the plus match. This generates a corresponding strip that aligns with the two X100 peaks, but it is the CB-CA-CONH spectra, which usually represents our I minus 1. So we know that this strip has a matching HNCACB with our X101. And the way that we will find the peak that corresponds with this is simply by putting our cursor through the middle of these peaks. And now we can see over on our NHSQC spectra on the right of my screen that it has intersected over a single peak, which you can see right here. So, knowing this, we will now select this peak on our NHSQC and assign it as our X101. I'm going to scroll over and we can see this one is our X101. And to get the HNCACB for this, we just ensure that the peak is selected and we add it just like we had for the X100. However, now you can see that there is a duplicated CBCA-CONH. You can simply delete one by selecting it and pressing the minus select button. Now we have our data, and from here you would do the exact same thing that we did for the past strips. I'm going to zoom out, and we can see that these two on our CBCA-CONH align with our X100, and it represents our I minus 1. So this is our 100. And you can see it aligns on our HNCACB perfectly with these two. However, you can see that the remaining two peaks are up here, which would represent the peaks for our X101. And now you just go through the process of assigning all of these. However, now since we are looking at the X101 on our NHSQC, you have to ensure that you change the nitrogen and the hydrogen accordingly. So now these are the X101. But since it aligns, this is our X100. And the red coloration symbolizes the carbon alpha. And you would simply go through and do this all over again. You can see that this one aligns with the green, just as I had over here. So this is definitely our carbon beta. Once again, we occasionally run into some peaks that weren't picked using apes. So to go over it again, we'll just use Control, press and hold the Control key, and press down, and it'll add a peak. Make sure that it is centered by pressing the two-letter code PC. These two remaining peaks are now known to be part of our X101. From here, you would follow the exact same steps as we had previously done and continued working forward in the sequence. I will be doing some off camera and showing you how to analyze our results. Now I have continued this process all the way to X103, and I've been doing the exact same thing, labeling it the same way as I had been at the beginning. And as you can see, the NHSQC is starting to be much more filled out. From here, I'm going to show you how to analyze these results. You need to type IR to open up the reference graphs. Since we are doing backbone assignment, you'll just need the one that has BB, standing for backbone. I'm going to close the extra windows. From here, it is really important to know some special amino acids. First, there is the threonine and the serine, which have very high carbon beta chemical shifts compared to other amino acids. Additionally, there is alanine, which has a very low carbon beta chemical shift compared to the other amino acids. Lastly, glycine has a very low carbon alpha chemical shift compared to the rest of the carbon alphas for the other amino acids. And additionally, glycine does not have a carbon beta chemical shift. Knowing these four special properties of these amino acids, it helps a lot in assigning within the sequence. To show this, I'm going to walk through X100 through X103. First, I'm going to look at X100 carbon alpha. As you can see, when I put the cursor over the peak, the horizontal line also appears on the reference chart. As I move up on the strip, it moves up and vice versa. So when I have it here, you can see the horizontal line goes through a lot of the chemical shifts for the carbon alphas of a lot of amino acids. So you really can't narrow it down much from the carbon alpha. However, when I go down to the X100 carbon beta labeled here, you can see that it is actually a lot higher than the other carbon alpha or carbon betas, excuse me. Here, you can see the carbon beta goes through the threonine chemical shift. However, for the sake of not over-assuming and also knowing that data can sometimes be slightly skewed, we are not going to say that it can't be serine. Threonine and serine have very similar chemical shifts, so for now, we can say that X100 is either threonine or serine. Now we're going to continue along. X101 carbon alpha very similarly goes through a lot of chemical shifts, so you can't say much. Now we look for the carbon beta, for X101, which is up here, and it goes through quite a few chemical shifts, so we really can't determine that one either. So for now, X101, we can just label it as a question mark. Next, 102, carbon alpha. It goes through quite a few again. Can't say much about that one. But the carbon beta, much like 100, is very high. And it even goes through threonine again. So, as a recap of what we know, X100 is either threonine or serine, X101 is a question mark, and X102 is a threonine or a serine again. So the possible combinations that we can see in our sequence are either threonine something threonine, threonine something serine, serine something serine, or serine something threonine. So, knowing this information, we're going to go over to our ubiquitin sequence. In here, as you can see, there are 4 places that this pattern occurs, with threonine something threonine, threonine something threonine, serine something threonine, or threonine something serine. So, how do we know which of these 4 places this part of the sequence belongs to? Well, we just have to continue going forward, which is why I found the X103. Here, the X103 carbon alpha is right here. As you can see, this is a much lower carbon alpha chemical shift than the rest of them down here, and it falls perfectly in the glycine. Additionally, as you can see here, there was actually no carbon beta found for this strip for the 103. Which, as I had stated earlier, is one of the properties of glycine. There is no carbon beta shift. So knowing this, we go back to the sequence and we look for where there's a glycine after the pattern. As you can see, that is right here. And when looking at the other 3 positions, none of them have a glycine show up afterwards. Knowing this, we can narrow down the X100 to X103 to be these 4 amino acids in the sequence. So X103 is actually a glycine 10. So knowing this, you can now go back to your NHSQC and reassign these. By pressing AT, you simply just change these to G10, standing for glycine, in the 10th position of the sequence. And now we can apply it. And you would just go through and fix all of these to correspond with the specific sequence of the protein until all of the residues have been assigned within the data. And that is how you manually assign the backbone.